Now we look at the second application of flow cytometry, which is widely used in cancer research, in cell biology. And the application is analysis of apoptosis by Annexin 5 PI flow cytometry. Now, before understanding the Annexin 5 and PI flow cytometry, we are going to discuss the simple steps of apoptosis and how the membrane structure changes during the pathway of apoptosis. So what is apoptosis? It is programmed cell death. Means it's a cellular mechanism where signaling is deciding that this particular cell is going to die. This is one of the method of cell death. So what happens if a cell is undergoing apoptosis? It occurs in 3 different steps. In first step, the chromatin compaction occurs. The DNA is getting compact and condensation of cytoplasm occurs. So the complete machinery of cells, including DNA, chromatin and cytoplasm, is getting compact. In second step, the breakup of nuclear envelope means nuclear envelope will be disintegrated, nucleus will be fragmented, DNA will be fragmented, and blabbing of cells occur, means small cellular bodies, or they are also called as apoptotic bodies, will be formed. And finally, these small apoptotic bodies will be phagocytosed by phagocytes or phagocytic cells, and ultimately cells will be disappeared. So healthy cell, it will start convoluting, chromatin will be compacted, cytoplasm will be compacted. After that, nuclear fragmentation will be there, DNA fragmentation will be there, and cell will be disintegrated with smaller apoptotic bodies which will be containing small fragments of DNA, and these smaller apoptotic bodies will be phagocytosed by phagocytic cells or phagocytes. Now, this is the macro explanation of apoptosis. Now we will do some micro analysis of what is happening in the cell membrane during apoptosis and how we can utilize that particular mechanism to understand the apoptosis is occurring or not by using flow cytometry. So if I look at the cell membrane of a normal cell, so a membrane is made up of lipid bilayer, or you can say phospholipids, and there is a particular phospholipid which is called as PS or phosphatidylserine. What is the speciality of phosphatidylserine? The speciality is, in normal healthy cells, the phosphatidylserine is present in the inner leaflet of membrane, or you can say towards cytoplasm, right? So if this is the plasma membrane or cytoplasmic membrane, this is outer part and this is inner part. The phosphatidylserine will be present in the inner leaflet, or you can say towards the cytoplasm. If the cell is entering into apoptosis, then the membrane starts flipping and this inner phosphatidylserine flips outside of the cell membrane. And if we are having presence of phosphatidylserine on the outer face of the plasma membrane, then we will be saying cell is undergoing apoptosis. And after some time, the cells will be dead because of apoptosis. So how it will look like? The membrane will be disintegrated. There will be big pores in the membrane, in the dead cell, and phosphatidylserine is outside of the cell. So during apoptosis, where the process of apoptosis is occurring, the phosphatidylserine will be outside. Healthy cell, phosphatidylserine will be inside. And dead cells, phosphatidylserine is outside with big pores or disintegrated membrane. This step-by-step manner of apoptosis is advantageous for us when we want to understand whether apoptosis is occurring or not. How? We are having a molecule which is called as Annexin 5, which we can label with a fluorescent dye. Let's say we have labeled the Annexin 5 with FITC, which gives us green fluorescence. FITC is the name of dye which is giving us green fluorescence. So we will take the cells, and these cells, in the single suspension of cells, single cell suspension, we will be adding Annexin 5, which is labeled with the fluorescent dye FITC. What happens? The Annexin 5 specifically binds to the phospholipid phosphatidylserine. So what will happen if a cell is healthy? Then the Annexin 5 will not bind, because all the phosphatidylserine are present in inner leaflet. So in a live cells or in a healthy cells, the Annexin 5 will not bind because phosphatidylserine is inside. And if I add propydium iodide, this will also not go inside the cells, because the cells are live, they will pump out the PI and we are not going to get the fluorescent signal. So, healthy cells, when you add Annexin 5 or you add the PI, you are not going to get any kind of signal, because the cells are live, phosphatidylserine is present inside the cell, there are no pores, so PI cannot enter inside, it will be pumped out. So no signal for Annexin, no signal for PI. That means cells are live. If cells are undergoing the apoptosis and they are still live, the apoptosis has started, but cells are still live, they are not dead. So what will happen? The phosphatidylserine will come out, Annexin 5 will start binding to it, and if the Annexin 5 is binding to it, it means we are going to get the fluorescent signal for Annexin 5. But still, the signal for PI or the propydium iodide will be absent. Reason? Cells are still alive, they will pump out the PI. So double negative means no signal for both the molecules, cells are live. Single positive means the signal for Annexin 5 is positive and signal for PI is negative, means cells are doing apoptosis, but they are still alive, they are not dead. But finally, in the dead cells, in the apoptotic bodies, there will be positive signal for Annexin 5 because phosphatidylserine is outside, and there will be positive signal for PI also because cells are dead, they will accumulate the PI, they will not pump out the PI and will start getting the PI signals. Can we do a virtual experiment to understand it? Yes. What we'll do? We'll take single cell suspension, we'll be adding Annexin 5 labeled with fluorescent FITC, we'll be adding the PI, we'll be taking the tube, putting into the flow cytometer, switching on, single cell array will be created, interaction with light will happen, and we are going to get this kind of graph, where on x-axis we are having the fluorescent signal for Annexin 5. On y-axis we are having the fluorescent signal for propydium iodide. What are these cells? Till now you have already understood. These cells, these dots, this population is negative for Annexin 5, negative for propydium iodide, means these are live healthy cells, not giving any kind of signal for Annexin 5 or propydium iodide. What are these cells? These are the cells positive for Annexin 5, negative for propydium iodide, means the apoptosis is going on. Cells have started the apoptosis, but they are still alive. They are not dead. We call them as early apoptotic cell. Positive signal for Annexin 5, but negative signal for PI. What are these cells? Positive for Annexin 5, positive for propydium iodide, means they are dead cells. They are dead because of apoptosis, or they are dead because of necrosis. We call them as late apoptotic or necrotic cells. Three types of population: double negative, live and healthy; Single positive for annexin PI, doing apoptosis but still not dead, early apoptotic cells. Double positive for both the dyes, both the fluorescent signals, they are late apoptotic, dead cells or necrotic cells. So this kind of graph you get when you are trying to understand whether the apoptosis is happening or not. Why we are doing this? Let's imagine again you are creating an anti-cancer drug. You're treating the cells, cancer cells, with anti-cancer drug, and you want to find out whether your drug is able to induce the apoptosis in the target cells or not. How you will do that? You will take the cells, culture them, treating with your drug, keeping it for 24 hours, 48 hours, 72 hours, taking out cells, adding annexin 5, adding PI, and getting this kind of graph where you can calculate the percentage of live cells, the percentage of early apoptotic cells, and the percentage of dead late apoptotic cells or necrotic cells. In real life, we can get this kind of graph. So what is this experiment? The cancer cells are treated with two concentrations of drug, lower concentration and higher concentration. After the treatment, annexin 5 and PI is added, and we have done the analysis on flow cytometer. BL1 is the fluorescent channel for annexin 5, BL2 is the fluorescent channel for PI. Most of the cells are double negative, means they are not positive for any of the fluorescent signal. It means these cells are live. You can see 94.30% cells are live cells. 4.06 have started the apoptosis without drug. It means even if you don't treat because of your procedure, cells are going to die. So this is the lower concentration of drug. You can see that cells have started migrating into this part of quadrant or this quadrant. These are annexin positive cells. So these are early apoptotic cells, 13.68. These are late apoptotic or dead cells, that is 7.27%. And here you can see with higher concentration treatment, many of the cells are going into early apoptosis and they are dying. You can see the percentage of live cell is what? Very, very less. Analysis of apoptosis by annexin 5 PI flow cytometry.